Good morning. A very hearty and warm welcome to you all in this popular talk series being organized by Department of Physics, Women's College, Agartala, Tripura. Before starting this auspicious occasion, I fold my hands before the light that brings prosperity, auspiciousness, good health, abundance of wealth, and destruction of the enemy's intellect. I fold my hands before the Lord, the maintainer of this creation, in the form of this light. I adore this light which destroys all the pains resulting from my omissions and commissions. Today, in this session, we are highly delighted by the kind presence of Srimati Manideepa Debbarma, Principal, Women's College Agartala, Tripura, as the president of this session. We have with us our respected teachers from various departments of the college. We are extremely delighted by the gracious presence of our resource person, the extremely talented speaker for the day. And last but not the least, we are more than overwhelmed by the presence of all the participants from across the country who have always cooperated with us from the very beginning. Once again, I heartily welcome you all in this session. Now, I would like to request Dr. Shubhrata Dev, Assistant Professor, HOD, Department of Physics, Women's College, Agartala, as well as the organizing secretary of this talk series to deliver the welcome address. Please, sir.
Am I audible?
Yes, sir.
Okay. Respected Principal Speaker, Dr. Chitra Mandal, SERB-Distinguished Fellow, Cancer Biology and Inflammatory Division, CSIR-Indian Institute of Chemical Biology, Kolkata. Respected President, Srimati Manideepa Debbarma, Principal, Women's College. Distinguished faculty members, dear colleagues, and my dear friends and students. A very good morning to all of you. It is a matter of immense pleasure that the Department of Physics Women's College is organizing this popular talk series, which is comprising of three lectures from renowned academicians, scientists in the month of January 2022. The first popular talk was held on 4th January 2022 and talk delivered by Dr. Sachin P. Nanavati, Senior Research Software Engineer, School of Chemistry, Cardiff University, UK. Today is the second one. As the organizing secretary of this talk series, I welcome our principal speaker, Dr. Chitra Mandal of IICB Kolkata, for her kind consent to be with us despite of her busy schedule. Thank you very much, madam.
Thank
Friends,
you.
as we know, due to the outbreak of coronavirus, commonly known as COVID-19, for the last 1.5 years or so, almost the whole world is passing through a severe crisis, and our nation is also not an exception. Both students and teachers were almost forced to stay at home during the peak of COVID-19 growth. That is the third wave, which is going on presently in our country as well as in our state. According to some statistics, in India, around 250 million students were affected due to school closures at the onset of lockdown induced by COVID-19, which included this pandemic posed several challenges in public and private schools, colleges, which included an expected rise in dropouts, learning losses, and increase in digital divide. The pandemic also called into question the readiness of the system, including teachers, to address such a crisis and sustainability of private schools and higher educational institutions. However, COVID-19 also acted as catalyst for digital adoption in school education and in college education. With schools and colleges reopening in many states, it is important that a careful strategy is built in to smoothen the transition of children back to school after more than 15 months or so. This transition has to consider the learning losses which had happened over the previous years, as well as to take a futuristic approach to build a resilient system which can withstand any future shocks. NEP 2020 and subsequent government initiatives such as National Digital Education Architecture, that is NDEAR, and National Institute Initiative for Proficiency in Reading with Understanding and Numeracy, that is Nipun Bharat, are expected to provide a blueprint for this transformation. Few months back, that is in the month of August or so, lockdown or suspension of classes were withdrawn. But after a peaceful few months, again, from the beginning of January 2022, rise of COVID-19 cases compelled the policymakers, governments to frame other thinking on offline classes. Already offline classes of pre-primary to 7th standard are stopped and their teaching lives are in online mode. From 8th standard onward, however, offline classes are advised with 50% students, but students are not attending the classes very regularly. Hence, to motivate the student community and also to provide them a glimpse of research and development in the field of science and technology, we have initiated this program. I can say that it is a program for young UG and PG level students, teachers, and researchers. I firmly believe that through this popular talk, young students, faculty members, and others will be able to listen to the resource person and upgrade their skills and knowledge about this burning issue. As an organizing secretary, it will be a request from my side towards the registered participants, especially to all my dear students, to please listen to resource person. Your active participation in this will definitely motivate us in near future to organize the same in some bigger platform. I must convey my gratitude towards Srimati Manitipa Devvarma, Principal Madam of Women's College, for her constant inspiration and motivation to all of us to organize such kind of program every now and then. I give special thanks to my colleagues, Dr. Sudip Devnath, Srimati Jaya Bhumik of my department, that is Department of Physics, for their kind support. I should also convey my sincere gratitude to Srimati Sharbarinath, Associate Professor and Secretary, Teachers Council, and Dr. K.V. Gita, Assistant Professor and Coordinator, IQAC, for their guidance. Finally, I must express appreciation to all esteemed members of the organizing committee for their support. It is also my duty to convey appreciation towards our technical coordinator, Srimati Pushparang Ranghav, Assistant Professor, Department of IT, for her help to organize this program successfully. Last but not the least, I am to express my sincere thanks to Ms. Ikshita Ghosh, Physics Honours student of 5th semester of our college, for running this program beautifully. Finally, I wish for the great success of this popular talk series, and I request all the participants, especially to all my dear students, again to listen to the renowned resource person and to put forward any questions which are creeping in their mind during the talk. And our resource person will be happy to deliver the answer of this question. So thank you once again. Have a good day. Thank you.
Thanks a lot, sir. Thanks for being so supportive. Now, the presidential address will be delivered by our respected principal, Madam Srimati Manidipa Deparma, Principal, Women's College, Agartala. Please, Madam.
Am I audible?
Yes, madam.
Good morning, respected Dr. Chitra Mandal, Distinguished Fellow Cancer Biology and Inflammatory Disorder Division, CSIR IICD Kolkata, today's resource person, other academicians, faculty members, my dear students, and my colleagues. It is my immense pleasure to welcome you all in this program on scientific contributions for the management of COVID-19 infection, organized by Department of Physics, Women's College, Agartala. We are extremely happy to have with us Dr. Chitra Mandal of IICB Kolkata as resource person. Madam, I, on behalf of Women's College and on my own behalf, welcome you in this program. I welcome all faculties, research scholars, and students from different parts of the country who are joining us through StreamYard. Our college was established in 1965 and it is the first college for women in Tripura imparting quality education exclusively for women. During this COVID-19 pandemic, we have organized many webinars, popular lectures on different issues, but today's talk has a special significance as we will be conducting this webinar particularly on COVID itself. For the last 2 years, we have been passing through a very challenging time due to the COVID-19 pandemic. The problem of COVID-19 is confined not only to our health hazard, but it has also multiple impacts on different aspects of our lives. This COVID pandemic is very serious as it spreads rapidly and mutates continuously. This disease can cause deaths and is responsible for the death of millions around the world. Initially, when the pandemic started, there was no means of controlling the infection or help in the process of recovery. But thanks to the advancement of science and technology and research, the virus was studied very soon and various vaccines were invented. Earlier, this process would have taken years, but now it was possible within a very short time. These vaccines are helping to save lives and avoid serious complications in most recipients. Not only in the invention of vaccines, research has been going on to study the viral characteristics, its mutations, infectivity, structure, etc., which in turn will help in designing drugs against the virus. In addition to this, various easy test kits have been designed and is readily available everywhere for quick detection of infection. Oxygen concentrator, pulse oximeter, ventilators are also being manufactured and widely available to bolster the health infrastructure. I hope that researchers who are working relentlessly on a cure for COVID in the shape of vaccines or medicines shall be able to relieve us from the serious pandemic in the coming days. We shall all be able to breathe again in the pandemic free world. With these few words, I once again would like to welcome Professor Chitra Mandal and other members, and I hope this program will be a very interesting and successful one. Thank you.
Thank you so much, madam. Thanks for always motivating us to do the best things. For the last 2 decades, it has been observed that there is a slow decline in the number of enrollment of students in undergraduate science subjects. It affects both quality as well as quantity of students pursuing higher education. This is primarily due to the shrinkage in the job and future scope of education and research. Conducting such a popular talk series will make a positive change in the mindset of both students and the teacher community. Main objective of this popular talk series are creating awareness among the students regarding possible scope of research in the field of physical science, highlighting the scope of future education to elucidate the various ongoing research topics, create a platform for the students to interact with the eminent persons in the field of science and technology, facilitate opportunities for networking, collaborations, and exchange of ideas with eminent academicians of the nation and abroad to boost young research and faculty members towards research and development. And now, this is the time for the much awaited session where the extremely talented research person will share her valuable words in this talk series. We have with us Professor Chitra Mandal, PhD, FTWAS, FNA, FASc, FNAsc, FNAMS, FAScT, SERB, Distinguished Fellow, Cancer Biology and Inflammatory Disorder Division, CSIR, Indian Institute of Chemical Biology, Kolkata, India. Before before starting this lecture, let me introduce Professor Mandal to the learned audiences. Professor Mandal is a SERB Distinguished Fellow, ex-Distinguished Biotechnology Research Professor DBT, Sir J.C. Bose Fellow DST. Madam completed her PhD from Indian Institute of Science, Bangalore, and postdoc from University of Pennsylvania, USA. Professor Mandal is having 170 publications, 15 patents, and 4 technology transfer. She has also supervised 30 PhD students. Professor Mandal's present research interests include identification of anti-cancer lead herbal molecules and potential targets, futuristic therapy for the development of an affordable healthcare for cancer, cancer cell and cancer stem cells. Highlights of educational qualification and professional career. Chitra Mandal, BSc and MSc in chemistry, obtained her PhD in bioorganic chemistry from Indian Institute of Science, Bangalore, and completed her postdoctoral studies on molecular immunology from University of Pennsylvania, USA. She joined CSIR, Indian Institute of Chemical Biology, Kolkata, as a junior scientist and ended her career as the director of this institute. She also worked as the outstanding scientist and head of the department. Subsequently, she was awarded Distinguished Biotechnology Research Professorship from DBT and also honored as a Sir J.C. Bose Fellow from DST. Currently, she is working as a SERB Distinguished Fellow. Dr. Mandal's notable contributions to biological and medical sciences has won her many laurels. She is an elected fellow of National Academy of Medical Sciences, India in 2013, elected fellow of the World Academy of Sciences 2012 as recognition of the outstanding contribution to science and its promotion in the developing world, recognized and appointed as outstanding scientist in biological science by the president in July 2010 to lead the CSIR Innovation Complex for Translation Research, elected fellow of the Indian National Science Academy, New Delhi, 2009, elected fellow of West Bengal Academy of Science and Technology, 2009, elected fellow of the Indian Academy of Sciences, Bangalore, 2006, elected fellow of the National Academy of Sciences, Allahabad, 2004, for the outstanding contributions in the field of medical sciences, both in applied and basic research, mainly in the field of glycoimmunology. She has been awarded the following: Women Scientist Award, CSIR Technology Award, Professor Bhim Shankar Trivedi Memorial Medal Lecture Award, Dr. Shubodh Mitra Memorial Orientation Lecture Award, Deshratna Dr. Rajendra Prasad Oration Lecture Award, Sir Ronald Ross Memorial Oration Lecture Award, Professor S.S. Katiyar Endowment Lecture Award, National Women Bioscientist Award, Dr. Yalla Pragada Subaro Memorial Lecture Award, J.C. Bose Fellowship Award by DST, Senior Scientist Oration Award by Indian Immunology Society, Biotech Product and Process Development and Commercialization Award, Shanika Oration Award by ICMR, Srimati Chandaben Mohan Bhai Patel Industrial Research Award, the P.B. Rama Rao Memorial Award. Her autobiography has been published in the book entitled Leelavati's Daughter, The Women Scientist of India by Indian Academy of Sciences, Bangalore. Today, we are very fortunate to have Madam with us. Please, Madam, platform is all yours.
Thank you. So I will go to directly to my slides.
Yes, ma'am.
Can you see the slides? Can you see the slides?
Madam, the screen is already shared, but the slides are not visible.
Can you see now? Is it seen? Can it be seen?
ম্যাডাম
দেখা যাচ্ছে?
ম্যাডাম স্লাইড গুলো দেখা যাচ্ছে না, বাট তোমার স্ক্রিনটা শেয়ার হয়েছে।
তাহলে what should I do? এখন দেখা যাচ্ছে? এখন দেখা যাচ্ছে?
হ্যাঁ ম্যাডাম, এখন ঠিক আছে।
আচ্ছা, so I will make the full screen. Okay. Is it can be seen?
Yes, yes, madam, perfect.
Okay. I am audible also, right?
Yes, madam.
Okay. So first of all, I would like to thank the organizing committee and especially Dr. Dev, who actually invited me, and I also would like to thank Principal Madam for her kind words. And this is an opportunity to share my views to the young budding scientists who are listening to me. And I must say that today after the lecture, at least if I can convince one student who will take his or her career as a scientist, I would be more than happy. So therefore, COVID, now because of the media and because we are going through a really hard time, almost everybody knows about more or less many things, many facts is known for the COVID-19 infection. However, today I thought I will share my views that what scientists' contribution for the management of COVID-19 infection. So rather I will tell you behind the screen, you will see the result in the market, but I will tell you the scientist hard work inside the lab, and so that you will also feel that yes, I can do it. If I become a scientist, I have a chance to save the world. Because you see, in this pandemic situation, one thing is very clear, that if you are a very rich man, lot of money in your bank or in your pocket, and if you are a very highly powered person like a politician, you have many powers, and but still in this situation nobody can help us. Only the scientist hard work inside the lab that actually helps to overcome such a tragic situation which we are going through. So this slide and this picture in the left side, it's very, very clear and very, very known to you. And this disease is severe acute respiratory syndrome, and this is coronavirus, so its abbreviation is SARS-CoV-2. So virus name is SARS-CoV-2, and the disease is COVID-19. Because of this virus, we get the disease called COVID-19, which is actually in Wuhan. They from China, December 2019, they declare they have a lot of patients with this severe acute respiratory syndrome. So by that time, they didn't know what is this virus about. So scientists within a month, they discovered the virus, which I'm going to tell you in details. How did they do it? So before doing that, let me tell you, so this is the outside of the virus, and red color thing is the spike protein. Actually, this looks like a corona, so therefore its name as coronavirus. It's a new strain of coronavirus. There are many coronavirus where earlier has been discovered, but this is a new strain, therefore it is written SARS-CoV-2. And right side of the slide, you can see the electron microscope photograph where you can really see the inside the cells there are viron. That means virus already entered into our cells, and some are budding viron, they are coming from the cells. So this is a real picture taken by the scientists to tell you that yes, this virus actually going inside our body through mouth and nose and infected our cells. So here, before I going deep into the story, I must tell you that this lecture has been organized by physics department. So a lot of physics students will be there. But I can tell you, although I'll be talking about more of biology, but I can rest assure you that without the physics background, chemistry background, computer background, IT background, all the backgrounds people, all the different streams, the people coming from different stream, as a hold their hand together and fought the battle. So if you are a physics student, don't worry, because we have many scientists who did his PhD in physics. They finally became a biologist. Myself, I did my PhD in chemistry, but afterwards I was so fascinated by biology, I shift my career to the biology. So don't worry about your background. Whatever you are reading, you just you just study that one. But later on, if your heart, you have to listen to your heart before you become a scientist. That scientist is a very tough job, because every day almost 90% of our experiment is failure, but 10% if it is a success, then that is a success, and that is a big success for a scientist. So this is just to encourage the student. Sometimes students think that, oh, I'm a physics, why should I learn the biology? So that is not the fact. We need a physicist, we need a chemist, we need all the scientists or all the students from their different background. Then only we can fight the battles together. So when a few years back, there was another virus emerged in China, that is severe acute respiratory syndrome called SARS. And within 4 months, scientists could identify the origin of this SARS. So they identify that it's a recombination event between a SARS-like coronavirus from bat and a coronavirus from pangolin. So there is a cross-species transmission. So therefore SARS, which is a family of the coronavirus, also came. That is the origin of this SARS. So that fact scientists identify within 4 months. And after a few years, there was another coronavirus came, that is MERS. The name is Middle East Respiratory Syndrome. So this again, scientists identify the fact that within 9 months, the MERS has emerged. So within 9 months, they find out that this is again cross-species transmission between bat SARS-like coronavirus and a camel coronavirus. But the SARS-CoV-2, that is the currently who is conquering our universe. So this SARS-CoV-2 virus, this is also a family of the coronavirus. Both SARS and MERS are the family of the coronavirus. And this is a new strain of coronavirus, scientists could not identify even after 2 years, since 2019, and now it is 22. But scientists with all sophisticated machinery, with all the background experience and knowledge, scientists till now could not identify any such cross-species transmission. So the question comes, is it a natural or is it a man-made? Some group of scientists think it is not natural. Why? Because if it is a natural like SARS or MERS, then it would have confined to a place, and then it will not spread like in all over the world, because it of course right now there are a lot of trafficking through the plane and other mode of communication, but still it can survive Antarctica as well as the very high temperature like Rajasthan and all. So these scientists believe that it is not natural. So if it is not natural, is it man-made? So then WHO, World Health Organization, they send a group to Wuhan from where it is origin. And Wuhan has a very big virology lab where not only Chinese investigator, but also many many brilliant virologists from USA, UK, French, Germany, they also work in Wuhan laboratory. So that is a very sophisticated laboratory. So they found that actually in Wuhan virology lab, virologists were working on this type of coronavirus, and probably there is a lab, from the lab, probably that is a leakage, knowingly or unknowingly, accidentally or not accidentally, we don't know, but definitely it is a man-made virus which is not natural. That I also believe so. So this is really a scientifically accurate atomic model. You can see very nicely that corona, which is a spike protein and which actually causing the biggest pandemic in 21st century. Since World War II, actually our world is really suffering this kind of devastation. So, and this each ball, if you can see minutely, each ball is an atom. So this, as you know, it is highly infectious and affecting millions of people and causing the disease called COVID-19. So please be remember, this is the virus name and the disease name. So when it is come out in 2019 in December, then WHO actually sent messages to all over the world, and they give the messages to the scientists that do something. And these are the black colored things I have written, that different topics all over the world scientists were working. They were trying to find out that what is the, how to prevent, how to get the prevention and get the control, and also social science people and COVID research ethics, all kinds of things. And finally they said the blue colored things has been already in knowledge, you know, the scientists based on their research, they recommended to WHO, and WHO asked all over the country that you should wear mask, you should hand wash, you should do the social distancing, you should do lockdown. So each and one preventive measure based on the scientific research, not like it is coming out of the air. So why social distancing and lockdown? Because scientists knew this virus is so dangerous. We have to cut down the transmission chain. We have to really break the transmission chain. Therefore, we need the lockdown. Containment zone we need, because so that virus cannot come out from that zone. And of course, if somebody is infected with the virus, you have to trace them and make it put it in a containment zone. And masking also is needed, because people knew that even you people can find out that one adult person in a day at least 20 to 25 times without knowing they touch their nose and mouth. So that is known. So therefore, if you have a mask, then you cannot, even if you by mistake, if you can put your hand towards the nose and mouth, it will be prevented by masking. And why hand washing? Next slide, I will tell you why hand washing is needed. Then scientists are trying to find out different drugs. So far, there is no specific drug against COVID-19. Scientists are working very hard, but there are some old drugs which is being using repurposing, which I will tell you a little later. And also a group of scientists try to find out the origin of the virus, because the virus is keep on changing um, their structure. And so already there are 5 varieties scientists have identified, that is alpha, beta, gamma, delta. And right now we are suffering from the Omicron mutant. I will tell you a little bit more about the Omicron later on. So then also the scientists are trying to find out if somebody has a pet animal like cat and minks, so whether COVID is spreading to their pets or not. So they found out so far cats and minks are susceptible to get this human disease, human virus. So in human, the transfer is human to human. That is very dangerous, because human to human, there is controlling that is very difficult. As you know, last 2 years we are suffering for that, but it is also spreading to cat and mink. And then I will talk about vaccine development. I will take a little bit more time on vaccine development, as well as I will tell you the story how scientists developed different diagnostic kit which we are successfully using for the diagnosis of this virus. So why mask? Because as I told you, that through the nose and mouth, because if I touch with any contaminated surface by hand, the virus will not enter through our skin cells. So it will enter only through nose and mouth by inhaling tiny droplet carrying virus. So you have to take care of the contaminated surface. Scientists with their research, they find out that how long that virus can stay on a wooden table versus a steel surface. All those things they actually did the research, and they know how long the virus can stay on different surfaces. So now I will tell you why you have to wash your hand with the soap. So this is the virus. As you know, I already talked about the spike protein, which is the corona-like structure. Besides that, there are several different proteins are there, because this virus is a mRNA virus. As you know, the virus definition of virus is it cannot survive by itself, because it doesn't have any genetic material inside the cell, unlike our human cells. So they have to enter the host cell, where this genome, in this case it is an RNA virus. So they actually enter into our body, enters into our cells, and this RNA will be released. And after RNA released in our cells, in our body, then they will hijack all the machinery from the host cell, and they nicely and happily they will replicate, and they will break that cells and go on replicating go on infecting the other cells. But here you see in the membrane, this RNA genetic material of this virus is actually very nicely coated with the inside a membrane. So this membrane has a lipid layer. So as you all know, being a student of science, the lipid doesn't dissolve in water. So if my hand is contaminated with the vaccine, if I wash with hand, the lipid will stay on and the virus will stay on. But scientists have found out that if you water with soap for 20 seconds, they have did the research and they found out for 20 seconds, if you keep the virus with the soap water, then this lipid will be dissolved and there will be a leakage. If there is a leakage, then all the RNA will come out and then there is no more virus. So you see, even simple hand washing advice to WHO came from a scientific research. So it is remarkably from the time of its discovery in December 2019 till today 2020, what our scientists have identified. Scientists, first of all, biggest success came when they identified the virus which is causing the disease called COVID-19. So why 19? Because it came out in 2019. So that is the disease called COVID-19. So they find out the causative virus of this disease called COVID-19. Why it is so important? Because if they don't know what is the cause of this disease, how do they find out the diagnostic? How do they develop the therapeutic? How do they develop the vaccine? So these are the reason that this is in my opinion, identifying the causative virus was the biggest challenge, and scientists very quickly find out, almost within a month, they find out that the SARS-CoV-2 is the causative virus for COVID-19. Until now, scientists are doing research, but because every few months there is a new variant, the coronavirus is changing its spike protein structure, and there is a mutation in the spike protein. Why it is important, I will tell you a little later. But you can see there are alpha, beta, gamma, delta, and now there is a Omicron. So by whole genome sequencing, they are every day tracking its variant by analyzing the sample from the patient from different country, because Omicron came from South Africa. So now, as I already told you, that preventing measure already, based on their research, they already tell the WHO within 2 months of their discovery of the virus. Then, of course, they design, once they know the structure of the virus, they could design, scientists could design and manufacture diagnostic and vaccine, which I'll be telling a little elaborate, so that you know how you know very well that we need the diagnostic, you know very well we need the vaccine. So what are the research they have done before we get proper diagnostic and proper vaccine? As I said, there is not, so far scientists did not successful to have a antiviral therapy, targeted antiviral therapy. So, but now good sign is they have developed some monoclonal antibody, but unfortunately this is very expensive, because to prepare a monoclonal antibody you need a lot of infrastructure, and this monoclonal antibody is against a single epitope. If this is a spike protein, and in the spike protein there is a lot of proteins are there, so it's against the single epitope this monoclonal antibody is prepared. The cost will be very high. It's almost 1 lakh per dose. Actually, in one vial there are 2 doses, but if you don't get 2 patients, then the patient has to bear the whole vial cost, which is 1 lakh rupees. Then total cost is 2 lakh for 1%. So that is the cost. So, but that monoclonal antibody worked for alpha, beta, gamma, delta, but not working for Omicron. Why, I will tell you a little later. So then time to time, scientists are keep on doing their research and keep on giving the advice to WHO, and WHO is giving the message to different government of different country. So as I said, I will mostly discuss with the diagnosis and vaccine, and a little bit of variants and a little bit of therapeutics. So before I go details about the diagnosis, you must know how this virus is attaching with our host cell. So angiotensin converting enzyme, this is a enzyme. In short, I will talk right now. It is A for angiotensin, C for converting, E for enzyme. So angiotensin converting enzyme, ACE2, unfortunately this enzyme present in almost all endothelial and epithelial cells. So it is a transmembrane protein. If you consider ACE2 as a log, and then spike protein of the virus, you can talk about the key. So spike protein actually of the virus binds to ACE2. So key binds to the log, and this virus, using a spike protein as a key, try to open the door with the binding to this log, try to open the door of the host cell. So this ACE2 is a very important protein on our host cell. It is a receptor for the virus. So ACE2 is the target for the virus, and as I said, to this target spike protein binds. So why it is so important? As I said, ACE2 receptor is found on most of the epithelial and endothelial cells. So it is on the upper and lower respiratory tract, and kidney, and part of the gastrointestinal tract, and mainly on the lung cells, pulmonary alveolar epithelial cells. So that is the reason, if you remember early 2020 or late 2020, when people in all over the world, people are dying due to the lungs irritation, lungs injury, because as these lung cells, epithelial cells, has a lot of ACE2 receptor, Therefore, what happened? This virus will be heavily loaded, heavily infected, infect the lung cell. So whole lungs will be full of virus, and there will be no oxygen passing through, and therefore people are dying just like that. So scientists have, with many hard work, they have find out this phenomena. And these ACE2 receptor are weakly present on the epithelial cell of oral and nasal. Therefore, virus is very clever. They don't want to stay in nose or mouth. They slowly move to the upper and lower respiratory tract and landed on the lungs. So lungs are the primary target of the SARS-CoV-2, and the spike glycoprotein of the virus binds to ACE receptor. So once this lock and key is happening, then virus is very, very easy. For virus is there is a membrane fusion of the virus to the host cell, and they release the viral RNA in the cytoplasm, and they nicely establish the infection. So once the viral RNA is in our cytoplasm of the human cells, then virus has got all the machinery to replicate. So virus hijack the human cells machinery and then replicate and propagate in the respiratory tract, and then invade and infect the lung cells. So this is just to pictorially what I said. This has been same thing. This is the virus. It is the spike protein. This is the RNA through ACE2 receptor. This is the host cell through ACE2 receptor. This virus goes inside the cell. There is a membrane fusion and inside the cells and release the viral RNA. And other protein also ACE2. This is like a Ghoreshchotri division. This protease actually what happened, these protease come to the spike protein here, and they will cleave and make the virus easy to come into the host cells. But in Omicron variety, what happened, this protease, they cannot cleave this virus. Therefore, in Omicron variety, we don't get the lung infection like Delta and Alpha, Beta, Gamma. So we get a lot of lung injury, because virus can successfully goes inside the cells and make a damage, and we get the acute respiratory disease syndrome. But Omicron variety, if you follow the COVID-19 disease from the medical report, the people are not getting lungs infection so much. So there are one report came, not more work is needed, but scientists, one paper I read where this says this protease in alpha, beta, and gamma delta variant, this was Ghoreshchotri division, but for Omicron, this is lacking. This the Omicron variety, the virus made a mutation in such a way that TMPRAS cannot bind here and cannot cut that, so that it can easily enter. So anyway, this is an infected virus. So infected virus produce lot of cytokine, and they activate the macrophage. So activated macrophage then migrate and go to the blood vessel, and thereby this virus goes to other organ. Wherever they found lot of ACE2, they bind and infect the cells. Now we'll come to the different diagnostic approach. So far what I told that this is the virus, and virus has a spike protein, and inside the virus there is an RNA. So once they get the swab sample from your nose and oral mouth, you get the virus. So from the virus, RNA has to be extracted, and from RNA reverse transcriptase, you have to make the DNA. So both RNA and DNA, by using this machine, thanks to all the engineer who made all this machine, and all the IT people who actually analyze the huge amount of data which come out from the machine, by this is called whole genome sequencing. It is a very expensive machine, but this actually machine, we can get the structure of RNA and structure of DNA. So within 1 month, using this machine, January 2020, within 1 month, scientists knew that what is the structure of the COVID-2. So using this RNA, I will talk about real-time PCR, RT-PCR. I am sure everybody knows about RT-PCR, but I will tell you the little science behind it. And then, of course, this once we infected with the COVID-2, then we get the antibody. So either you can detect the antibody or you can detect the antigen, And by all this method, you know whether you are infected with this virus or not. So one by one, I will tell you the little scientific story behind all these diagnostics. So you have to, of course, clinical symptom, doctor will tell you how much fever is there, whether you have a 90%, 98% oxygen or not, all those things. But today I will be talking about RT-PCR. That is the gold standard for diagnosis of COVID. So what is the abbreviation? It is real time, or sometimes it is called reverse transcriptase polymerase chain reaction. It is a very powerful tool to, and this machine actually almost every research laboratory has that. Without this machine, we cannot do any experiment. And during this 2020, from our laboratory, we actually donate many RT-PCR machine to the hospital. We send our research scholar to train their technician how to do the polymerase chain reaction, because in the initial phase, technician in the hospital, they don't know how to do the polymerase chain reaction, because RT-PCR only used in the laboratory, not in the hospital. But because of the pandemic situation, we had to transfer our machine to the hospital. And this machine, this polymerase chain reaction is a very, very important reaction, as it is so important that back in 1993, that American biochemist Kerry Banks got the Nobel Prize in chemistry along with Michael Smith. So what does it do? This polymerase chain reaction, it actually detects the genetic material. What is the genetic material for our SARS-CoV-2? It is an RNA. So they detect the RNA. And result we come usually, that we know, within 24 hours we get the result. Somebody will come to your house and they will take the swab sample from your nose or mouth and then take to the lab where this machine is there, and then they do the following reaction. So this is the principle of the RT-PCR. I know many people probably know, but I thought that as a different stream of people are there, Dr. Dev told me that there will be different kind of people who will be attending this seminar. So I thought I will talk a little bit about the RT-PCR, which is so known. The name is so familiar to everybody nowadays in the whole world, even in our country. So what is that? So from virus we get the RNA. So RNA, we need to make the DNA, and then that DNA we have to do the amplification. So when we collect the sample, there are very small amount of this genetic material. It's such a tiny bit that we cannot detect. So it has to amplify. Therefore, PCR, polymerase chain reaction, by which you can do the amplification. How did it amplify? There are 3 steps: denaturation, annealing, and elongation. I don't know if you can see my hand or not. If you can see my hand, you can look at my hand. So as if these 2 hands is the DNA double stranded. As you know, our DNA is double stranded. So this 94 degrees centigrade, if you heat the sample for certain time, this double stranded DNA will break down to single stranded. Now this single stranded DNA will act as a template. So now during the annealing step, when you lower the little bit of temperature for certain time, and in the reaction mixture, you have given the primer, forward primer and reverse primer, then these primer will bind to the complementary sequence. So my left hand will bind to some primer, my right hand will bind to some primer. So you will get, then you elongate by raising the temperature in presence of Taq polymerase. So after some times you get a double stranded DNA. So from one cycle of denaturation, annealing and elongation, from one double stranded DNA, you get 2 double stranded DNA. So these steps has to be repeat for 20 to 40 times. Then you will get enough DNA that can be detected by the machine. So here the sample collection, you get the RNA. From the RNA, using reverse transcription enzyme, you get the DNA, that is the double stranded DNA. Using the DNA polymerase and the primer, you need to amplify this product, and there we use a fluorescence probe, so that machine will give you the fluorescence intensity. So when the fluorescence intensity, so as you repeat the cycle, these 3 steps, as you repeat the cycle, you slowly started to seeing the fluorescence intensity. So if you have done RT-PCR anytime in your friends or you by yourself, because it is needed even if you want to see a doctor. Doctor says that bring your RT-PCR data, then only I can touch you or I can check you like that. So you go to anywhere, you need an RT-PCR negative report. So in that report, you see there is a CT value. So what is the CT value? CT value is the number of cycle after which fluorescence of the PCR product can be detected. As I was telling, you started with a very small amount of sample collected from your nose, then that product you need to amplify. So to start with, if you have a lot of virus, then after a very few cycle, you will started seeing your fluorescence intensity. So if your CT value is low, that indicate you have a very higher viral load. So that probably you can now understand that if you were to start with, if you have a very low amount of DNA, to amplify in a certain amount to see the fluorescent intensity, you have to do many cycles. So scientists had given some table to the doctor so that doctor can interpret the RT-PCR data. They suggested that if the less than 25 is the CT value, then patient has likely to develop severe outcome because the virus load is too much. If it is 25 to 30, then the patient need to be monitored. If it is more than 30, then there is a complication, because either you are early infection where virus load is very low, or you are in the later phase of infection when virus has been cleared but leaving behind little bit of genomic content. So this is the RT-PCR principle, and simply by looking at the CT value, doctor will decide whether the patient has to be taken to the ICU or not. So that is a gold standard diagnosis. Now I will talk about another amazing technology that has been done by these two ladies, and it is a women's college. So I am proudly make this slide for you people. These two ladies got the Nobel Prize in 2020. And for what they got the Nobel Prize? They developed a CRISPR-Cas9 system, which is called gene editing technology. Can you believe gene editing? Don't you feel thrilled that your gene could be edited? And this technology actually make the world a different world. Like people now can think about that if I have a cancer, I know the breast cancer, for example, the scientists know which gene is mutated. So by using this system, using their discovery, they can actually now think about that taking out the cells, cut that bad mutated gene and put it back the good cell into the thing. So this is like a really a centuries important discovery. Of course important, therefore these two ladies has got the Nobel Prize. But you probably wanted to know from me, madam, what is the CRISPR-Cas? CRISPR-Cas is slowly, I don't know whether, I know if you, I cannot see you, so I don't know your face, whether you are understanding or not. I will try my best to tell you in a very simple way what is CRISPR-Cas, because you must know this is only 2020 Nobel Prize. So a lot of things can be done. You are, as a young scientist, budding scientist, you must know and you must think something that when you will become a scientist, whether you can be involved in gene editing technology. So it is a C for cluster, R for regularly, I for interspace, S for short, P for palindromic. Palindromic, you know, palindromic number, physics student at least know. From left side or right side, which side you read, it read the same thing. Like 1 to 1, from both left and right, you can see 1 to 1. So like madam, both way, left side or right side, if you tell madam, it's a madam. So that kind of word is called number, or word is called palindromic. So that means this cluster, this CRISPR is a short repeating DNA sequence with a spacer sitting in between them. So suppose if you can see my hand, suppose I am wearing 20 bangle, and 10 bangle is a red color, which is the short repeating DNA sequence. But I am wearing 20 bangle, right? So after the red bangle, which is the repeating DNA sequence, short repeating DNA sequence, after that the blue bangle, that is the spacer. After the red bangle, again the short repeating DNA sequence, again the yellow bangle, that is a spacer. So like that, it is a cluster regulatory interspace spot repeat. That will be repeat. This short sequence will be repeat by intervening between the spacer. So that is called CRISPR. So what is Cas? Cas is an enzyme which is a Cas CRISPR associated enzyme. There are 9, 10, 11, many Cas9 is there. So why, how, so you might ask me the question, how this Professor Dudna, how he think, what is the importance of CRISPR? Suddenly, how did he get this sequence? So she was working with a bacteria, and many other scientists also, I must mention, their contribution also is enormous in this finding. So they found that almost all the bacteria who are surviving millions and millions of years before human being, how do they survive? So all the bacteria, almost all the bacteria has this CRISPR sequence. So she was thinking that why all the bacteria has a CRISPR sequence? They must have some benefit. Then she found out that is the bacterial immune system. That is, using this sequence, they actually protect from the virus infection. Just like human body has a lot of immune cells, T cell, B cell, from which we are every day we are encountered by so many infectious diseases, but not every day we are getting sick because of our immunity power, because of our immune cells. So for bacteria, they have a CRISPR sequence by which they can survive the attack from the virus. How do they survive? So they find out that before the CRISPR, the bacteria genome has different CRISPR associated proteinase, that is an enzyme. So Cas, you can think about a scissor. So scissor will cut something. What it cut? Because when the virus is attacking the bacteria, they bind to the CRISPR sequence. So what happened? This enzyme Cas will, as soon as they got a sense that virus has attached to the bacteria, this crystal like an hairpin, we have a hairpin, so Cas will move to the CRISPR and then it will cut the CRISPR sequence. So these CRISPR-Cas together make a CRISPR basis of the technology known as CRISPR-Cas that can be used to edit the gene. So why I am taking this CRISPR-Cas story here? Because lot of scientists now using this CRISPR-Cas system and try to develop the sequence, try to develop a diagnostic. So this is the Cas9, CRISPR-Cas9 diagnostic. But before that, there was Cas12, Cas13. The name of the diagnostic was Sherlock and Detective. But today I'll be talking about Feluda test. Why Feluda? Because you know Satyajit Ray's movie, right? Feluda is our hero. So this guy who discovered this Feluda test is a Bengali guy working in New Delhi. His name is Shobhik Maiti. His laboratory actually very carefully select a Cas enzyme from a bacteria, because all the bacteria has CRISPR-Cas system. So from a bacteria, he carefully select a Cas9 enzyme, which can actually sequence, which can actually distinguish between a nucleotide sequence differing by only one mismatch. That means everyday virus is there is a variant. So by his diagnostic, we can this Cas can distinguish from one variant to other also. So this is the Feluda test, which is the first CRISPR-Cas9. As I said, Cas12 and Cas13. Using Cas12 and Cas13, there are two more tests are available. But Shobhik Shobhik Feluda test is the first CRISPR-Cas9 rapid test available for COVID-19 detection by lateral flow assay. What is the advantage? You don't need a PCR machine. You don't have to go to a lab to give your sample. So although it detects the genomic sequence, which RT-PCR also detects the genomic sequence, but in Feluda test, you don't need to do the need to have a RT-PCR machine, and you can do it at home, and result will be within 45 minutes compared to PCR, which takes 24 hours. And the result is comparable to our RT-PCR result, and sensitivity and specificity also is very high. So it is by Tata Group is already started marketing this Feluda test. So you see how the Nobel Prize work in 2020 and gives the vaccine, gives the diagnostic in 2021. So besides that genomic detection, we can actually detect the sample and also detect the virus antigen, and you can take the blood and also you can detect the antibody. how we can detect? This is a lateral flow immunoassay. This is a nitrocellulose piece of paper, just like pregnancy test. This is a piece of paper, and there is a sample load. You collect the blood, collect the swab sample from your nose at home, and this is actually available as a home kit. You can go to the pharmacy, you can buy this kit, and you can do it at home. So what you do, you collect your sample from your nose and put the sample here, that is the antigen, and then you add your already in conjugate paired, that you already antibody against this virus with the colloidal gold is there, and these actually migrate from bottom to top. So these will bind to this will move to this place, and then virus will bind to this antibody, then it is moved, and where in test line the another antibody which can bind to the virus that is already attached and in the strip, and once they reach this complex, when it reach there, it is precipitated and you can see a line, and that's how you know whether antigen is in your nose sample, whether virus antigen is present or not. And this is a very quick, within 15 to 30 minutes, sitting at home, you can do this test. No lab infrastructure is needed, but it is not so accurate like your PCR test. Similarly, you can collect your blood, and blood sample has lot of antibody, IgG antibody and IgM antibody. You put into the sample pad, and then in the conjugate pad already there is an antigen attached with the colloidal gold is there. So when this antibody moves to this section, it will bind to the antigen virus antigen. Then when this complex moves to IgM line, there is already an IgM antibody which binds to the Fc portion of this antibody, and you will see a line. So whether you know whether it is an IgM line or whether this is an IgG line. When you take the vaccine, within 5 to 10 days, you get within 10 days, you get the IgM, and then after 2 weeks or so, you get the IgG, which stays for 5 to 6 months. So by this lateral flow immunoassay is very important to detect both antigen as well as the antibody. And there are ELISA also. And so I talk about 3 different assays, taking the genetic material, that is RNA by RT-PCR, taking the protein, the virus protein, and bilateral immunoassay, and also the antibody bilateral immunoassay, so that by using your viral antigen, you can detect. So these are the 3 different principles by which the diagnosis has been made by the scientists, and there are many diagnoses are available right now throughout the country. Not in our country, but many different countries. So now coming to the vaccine. Why vaccine? Because there were no effective drug as there, only repurposed drugs are available. And although we are taking the preventive measure by wearing mask and etc., but in such scenario, we have to get the vaccine. So scientists from day one, they put all their effort to develop the vaccine. And another reason that previously against the deadly disease like smallpox, polio, measles, hepatitis, we had a success by utilizing the vaccine. So vaccine prevents from us from all these infectious diseases. And also more importantly, who are comorbidity, like high blood pressure, diabetic, they are usually immunocompromised. So even immunocompromised and unvaccinated people also will be protected if we immunize more than 70 to 80% of the population. Then we'll get the herd immunity. And once we get the herd immunity, we can protect even a few unvaccinated or comorbidity patient. This picture is very familiar to all of you, like this gentleman, because if you talk about the vaccine, I'm sure this is a very familiar picture. You know this gentleman. He is an English doctor named Edward Jenner. He, back in 1796, almost 200 years ago, he found out, that time no concept of vaccine. So, but his intelligence, through his intelligence, he found out that in the cow smallpox, that গোয়ালিনী যারা cow নিয়ে কাজ করে, they don't have that disease, that smallpox. So he realized that maybe এই যারা কাজ করে গরু নিয়ে, তারা হয়তো somehow এদের ব্লিস্টারের থ্রু দিয়ে তাদের শরীরে হয়তো কিছু ব্লিস্টার ঢুকে পড়েছে, সেই জন্য হয়তো তারা প্রোটেক্টেড। With this faint idea, what he did, Edward, what he did, he actually, a 9-year-old boy who actually, the son of his gardener, he injected from the cow smallpox blister to his hand and then waited for a few months and then exposed him to the real virus, and he found to his astonishment that he never developed the smallpox. Of course, nowadays you cannot think about this kind of experiment because there are a lot of regulatory, lot of human ethics are there. You cannot inject anybody without any permission. Now come the Louis Pasteur. He also give us different vaccines, cholera, anthrax, and rabies. Therefore, we are safe from this disease. But how the scientists make a vaccine within a very short time? Because of this previous history, which I just now talk about, different kind, all those vaccine has been created almost 10 to 15 years of rigorous research. But world doesn't have, world cannot wait so many years, because by the time, because the way the COVID vaccine, COVID virus is, SARS-CoV-2 virus is spreading, it's really notorious. So we have to get a quick vaccine. So then you can ask me a question, why it takes 10 to 15 years? Because first of all, scientist has to do extensive laboratory research to find out a proper antigen against which the vaccine can be made. So to find out a proper antigen, it's a huge job, very big job. So after that, you have to do the preclinical, then phase 1, phase 2, then phase 3, then only you get a vaccine. So you can ask me, what are those phase 1, preclinical and pre-2? This slide I kept you because I do it for my cancer drug. I do it regularly in my lab. So I thought I will share the fun of the work in the lab with you people. So preclinical investigation means in the laboratory, scientist has to find out what antigen is their candidate. So by September 2020, not September, I think by July 2020, almost within 6 months, the to WHO, all over the scientists from all over the country, 200 antigen as a candidate for the vaccine has been submitted for the clearance from WHO. So you see, there are a lot of variation in the antigen. So whoever made the proper antigen selection, their vaccine will be successful. So antigen you have to take from where? From spike protein, because spike protein is the target through which SE2, which is binding to the human cells. So all the scientists from different country, they select the spike protein, but spike protein is a huge protein. So which portion of the spike protein they will take? They have to take a very tiny bit of antigen, tiny bit of protein. So once they identify that antigen, they had to inject into the animal. So let's say in this case, we use mice. So in that mice, you have to inject very, very high dose, and to find out that at what dose the animal are not behaving normal. So we inject group of different mice and injected different doses. One is 1 milligram, 10 milligram, 100 milligram, 1 gram, like that. And then after a few days, we dissect the mice and check their lung, liver, heart, kidney, and blood parameter and everything. So by this preclinical and investigation, we know the dosage, and then we need to go for the approval from DCGI. This is from India, Drug Control of Government of India. This is from USA, Federal Drug Administration. So once we need to get the approval to go to the human being. So this is called phase 1. In phase 1, you have to select a very healthy, normal individual, no comorbidity. And let's say 100 individual is the volunteer. So 50 will be given the dose, the highest dose which animal can bear. To start with highest dose, you have to go down. That what is the minimum doses, you get a better efficacy. So you get the different doses to this human, and then you check their efficacy, that whether they are protected from the virus or not. So once you pass the phase 1, and of course side effect. Once you pass the phase 1, then you go to the phase 2, where you have to go to a very large number of human being healthy. Here, not only you have to select not only the healthy human, you have to check the comorbidity patient, you have to take gender, female, male, all the gender, all races. So demographic population, you have to go in the phase 2 clinical trial. And then with the selected dose, selected from the phase 1, you have to administer at that dose in 500 of these people, and 500 people will get the placebo, and then check their side effect and efficacy, of course efficacy. Once, if you are lucky, if you get the phase 2, then you have to go to the phase 3, where you have to take 10,000 to 60,000 people in this population, and you have to really check the effectivity of the vaccine. You have to check very carefully the symptom, their antibody level, their virus level by RT-PCR and all these things. And then if you pass the phase 3, then only you have in your hand the specific vaccine. But this is the line I make you. This is the traditional way, the vaccine development time. As I said, it almost take 10 to 15 years, because once you pass the phase 3, after that also there are a lot of steps. You have to have enough infrastructure to manufacture that vaccine in a large quantity. We are fortunate that India is the first vaccine. India is number 1 actually in terms of vaccine purification infrastructure, because we are really make lot of vaccines. So our infrastructure is very strong, number 1 in the whole world. So once you manufacture, then you have to distribute. Some of the virus is very temperature sensitive, so distribution is a problem. So all this takes a long time. But what happened for the COVID? They did not get clinical proper ethical clearance. They gave accelerated vaccine development time, and they get the emergency permission to go for phase preclinical to the phase 3 trial. So you can ask me that how did the scientists within a year develop COVID vaccine so quickly? Because that, as I told you about the SARS and MERS, these are the family of the coronavirus. So before COVID came, there was a lot of infection in China. It was confined only in China, both SARS and MERS. So there, what happened, scientists were working with this virus, so they know how to deal with the corona type of virus. So because they are also the same type of coronavirus. So once the disease identified in December 2009 in Wuhan city of China, by 1 month, using that hi-fi sequencer, scientists already finished the genome sequencing. And once they reveal the genome sequencing, they immediately know it is a coronavirus family, and they name as SARS-CoV-2, and they find out this is the cause of the COVID-19. As I said, without knowing the cause, it will be very difficult to make the diagnostic as well as the vaccine. So by January, they know the data on the structure, they know the life cycle. So that was established within 2 months from December 2019. So this is fantastic. So scientists all over the world working day and night to fight for this disease, fight to understand the virus. And by that time, they also know the importance of the spike protein. As I told you, spike protein is like a key. So they know the spike protein is responsible for the attachment to our host cell, for the membrane fusion, and finally to the entry. That was also established very quickly. So as I said, spike protein, once they identify the spike protein as a target, they immediately use this target protein for both for the vaccine and for the antibody development for doing the diagnosis. So now you have the vaccine, but you need to vaccine. How do you inject? Because it's a piece of DNA, piece of RNA, piece of whatever, the gene portion. How did you inject in our body? You need a vehicle, because when you go from somewhere, you have to enter into a train. So it's like you need a train by which you put the spike protein target inside the train, and you ask the train to go into our host cell. So here, when they were developing the MERS, that is another type of coronavirus, when they were developing the MERS vaccine, they knew that adenovirus, which is another virus which is harmless to our human body. So remove the material from the adenovirus, and they put the vaccine material into the adenovirus, and they use adenovirus as a vehicle to inject their genetic material into our body. So this is really a worldwide cooperation led to a first track research and the clinical trial. So besides that, USA government gave huge funding because you need money to do the research. So they gave huge funding to the public sector and all the researcher to accelerate the process. So therefore, by December 11, 2020, exactly 1 year after the COVID came, the both Pfizer and Moderna, those are the two USA company, very big company, and for their huge fund and for their experience, especially Moderna was for a long time they were working with the RNA vaccine for different disease, but they were not getting any clearance from the FDA because people were very skeptical that foreign DNA will come to our DNA, our genetic material. People didn't like it. So FDA for a long time did not give any ethical clearance for their earlier research. But now the situation is different. FDA was forced to give an emergency use authorization to this company, giant company, Pfizer and Moderna, for their vaccine. So this is the world first mRNA vaccine, first genetic vaccine, you can tell. This is the first genetic vaccine which has received the approval, of course, emergency approval from the FDA without clinical trial, without proper human clinical trial. But we are very fortunate of the scientific research and scientific talent. Because of their scientific foresee, this COVID-19 mRNA vaccine was very effective in preventing the hospitalization. So this fast-track vaccine development within a year is really a very big contribution from the scientists. So for Pfizer, Moderna, what happened? They did not use adenovirus. They use a lipid nanoparticle as a vehicle. So lipid nanoparticle, this messenger RNA of the spike protein of RNA, they wrapped in a coating called lipid nanoparticle for the delivery to our host cells. So what will happen once it's delivered to the host cells? This messenger RNA will be released and give the instruction. As you know, RNA, messenger RNA will sit on the ribosome and protein will synthesize. That is the central dogma, DNA to RNA to protein. So mRNA will translate to the protein on the ribosome and asks the instruction to the cells that to produce harmless spike protein. It is spike protein, but it is not attached to the virus. So this protein is harmless to our body. So they make tens and thousands of the copy, and once they make all these copy, they will destroy the genetic material of the vaccine, and they will instruct our body's immune system will see this spike protein as a foreign protein, and our immune system will be activated, and they will produce a lot of antibody against the spike protein. So that is the idea of the vaccine. And once our immune cells is trained to see this spike protein, so in actual scenario, later on when we get the virus infection, our immune system already know how to fight with this spike protein. So they will fight and the virus will be killed. So this slide I kept you for one reason, because I told you the structure of the virus, RNA, and the spike protein. And I also told you that spike protein bind to the ACE receptor, but I did not show you how the ACE protein looks like. It is a very huge protein. So scientists has a tough job to find out. They knew that spike protein binds to ACE receptor binding domain, but they had, as I was telling, we need a very tiny bit of RNA from this spike protein. So they had to exactly know where it is binding. That they use a cryo-electron microscope. I don't know whether you know the name of the thing. Thanks to the physicist, thanks to the engineer, they made this cryo-electron microscope. Using this electron microscope, cryo-electron microscope, scientists now can freeze at 180 degrees centigrade, can freeze each and every frame-wise the binding picture of the spike protein to the ACE2 binding. Now come the computer expert. Huge IT specialists came together, put their head together and analyze those different snaps of binding of spike protein to the receptor and analyze that which protein is really will be good to bind of the spike protein to the receptor binding domain of the host cell in the ACE2 receptor. So that's therefore you get the RNA vaccine, you get the DNA vaccine, you prepare the protein, so you get the protein vaccine. And then this DNA, you can put it into the adenovirus, so you call vector vaccine, and you get the inactivated vaccine means this whole virus, it can be treated with some chemical so that virulence will be inactivated, and then whole virus we can use as a vaccine. That is the vaccine Bharat Biotech in India is making, and name is the Covaxin. We are getting, many of us get the vaccine, this inactivated virus. And Covishield is the vector vaccine in which adenovirus was used to put the DNA into the virus. So whoever take the Covishield, they take the vector vaccine. Of course, they think the AstraZeneca who prepared this vector vaccine, they told that this genetic material once inside our nucleus, they will not incorporate it in our DNA. But tell you the truth, it is my own personal opinion. I will be very happy to get the mRNA vaccine, because mRNA vaccine, I know there is no way it will be integrated in my gene system. But as I said, mRNA vaccine needs very low temperature transportation. Therefore, our India government did not purchase any mRNA vaccine from Pfizer or Moderna from the USA. So these are the adenovirus. Already I told you the Covishield. This is the adenovirus replication deficient viral vector they use. So we get the spike protein DNA into our cell. And again, like last time I told you, the DNA will instruct our instruction to give our instruction that to make a huge number of spike protein. And as usual, like I told you before, our body immune system will work. Covaxin, already I told you, this is an inactivated virus, inactivated vaccine. But all these vaccines did not get a proper clearance. They received only the emergency approval for Government of India regulatory authorities. So as I told you, that whole world put their hand together. So this slide just I like to mention that for Pfizer, both USA and Germany, they make this RNA vaccine. Moderna make the RNA vaccine. Viral vector, Johnson & Johnson make the viral vector vaccine, means DNA particle inside a virus particle, a virus coating. And then AstraZeneca, UK and Swedish company, they, along with our Serum Institute India, they make the Covishield, which we do get. And Sputnik is the Russian vaccine. And China has made 3 vaccine, 1, 2, and 3, but nobody believed their vaccine. And inactivated vaccine is Covaxin, which Bharat Biotech is making. Protein subunit, very recently it has come, Novax, and people, it is not yet used in very rightly. And DNA vaccine, very just last month in December, Zydus Cadila India make the DNA vaccine, that is a fast plasmid DNA vaccine. All the vaccine is more or less 2 times you have to take. Now, of course, they are telling that you have to take the booster also. All are intramuscular. And these are the country they are using this vaccine. And I wish our country's name is there in case of Moderna or Pfizer because of this genetic material. So Zydus already I told you. So vaccine already I told you that how vaccine will be working. They will deliver DNA or RNA, whatever entering into our body through vaccine. They will produce the harmless protein of the COVID-19. Now this protein, our immune system will see antigen presenting cells, that is the macrophage, will first see the viral protein, and they will instruct the T helper cell. They will make the 2 type of immunity, cell mediated immunity and humoral immunity. They instruct the T helper cell to make the B cells committed to produce the plasma cell, which will produce the antibody. And once the antibody is produced, these B cells will keep as a memory cells. Once we really will be infected by the virus, this memory cell will remember the spike cells and they will come and kill the virus. So we will not get, cell will not get affected by the virus. And when these antigen presenting cells give the information about the spike protein to the cytotoxic T lymphocyte, this T lymphocyte will be activated and they will lyse the virus infected cells, and that is called the cell mediated immunity. So some important vaccine that some people in foreign country, they are not taking vaccine. That's how in USA, Germany, many unvaccinated people are there. Although they have a lot of vaccine in hand, but people are reluctant to take vaccine. Maybe due to the afraid that one they will get the vaccine, they will get the COVID-19. But as an immunologist, I can assure you, if you take the vaccine, you will not get any COVID-19 from the vaccine. You maybe later on infected with the vaccine, but your immune cell will take care of that. And vaccine will not alter or damage the genetic information. This is I am telling based on their company's leaflet. And however, even if you take the vaccine, you must maintain the all the preventive measures like mask, wash hand, and etc. I told about herd immunity. So herd immunity is like that. If there is no vaccination, all the people will be infected very quickly because it is a human-to-human transmission. But if you get more immunity, then unvaccinated people also will be protected. And so if you make a contained area, then these infected people, infected man will not get, will have a less chance to infect the blue color which are unvaccinated. So idea is that 80 to 90% of the people should be vaccinated so that we get the herd immunity, which is very, very difficult. The vast country like us and poor country like Africa, because they not even 10% of the African people got the vaccine. So many unvaccinated people are all over the world, and now world is very small because of the journey, lot of plane, lot of people, lot of trafficking is there. So due to the trafficking, the virus is moving from one country to other. So as I told you about the variant, already 5 variants is there. Scientists have identified 5 variants, and those variants, there are many other variants, but these are the 5 variants which is considered to be variant of concern because they make the havoc changes in our human population. So, for example, Delta is twice dangerous than the earlier Alpha and Beta. Omicron is less dangerous. It is spread more easily than other, but Omicron causes less severe disease. But recent paper is coming that we don't have any chance to be happy because Omicron is doing something to our immunological system, so that people after Omicron is over, Omicron wave is over, people are going back to the hospital, and if you listen to the news, you see death rate is not decreasing. That means Omicron, although it is less virulent, it is doing something. Scientists has to do the research more on Omicron and see what happens. So we have to wait for the scientific investigation. So why Omicron is not so dangerous to the lungs? I told you before, but I made a special slide for you to understand the scientist contribution or to understand the actual mechanism, because new Omicron variety has a 30 mutation where in the spike protein, which is the key of the virus. So then these 15 of the 30 mutation actually in the receptor binding domain. Do you remember that ACE2? I showed you the receptor binding domain by which scientists work with the cryo-electron microscopy. So because of this mutation, actually some of the enzyme is not binding proper. This binding is very tight, so some of the enzyme is not working to keep the virus inside the lung cells. So that's how Omicron is, although it is a variant of concern, but it is not affecting the lungs. So this is in a positive note. One Israel scientist in his tweet, he made this. I like this tweet, so I put a slide on that. But the summary of the slide is that Omicron, according to this doctor, Israeli doctor, Omicron is literally the vaccine that vaccine company could not make. Because with this Omicron, once we get the Omicron infection, it is I think it is an attenuated vaccine because it is not giving any lung injury or any severe injury. So no hospitalization, no critical patient, no oxygen is needed, no incubation, and everyone, if is exposed, will get it. And so herd immunity will get it without even giving artificial immunity. So he thinks that we must show the gratitude to the nature for finally eliminating a man-made lab-leaked nasty virus, and we are blessed. I hope this doctor is right. We need to see in the future that what happens. And I am almost end of my talk. This is just to show you in India, we have already reached the first wave in August 2020, and we are waiting for, we already experienced the second wave in June 2021. And we are waiting for the third wave or third wave. Is it third wave is coming or not? This is the death. As you can see, in the first wave, because the urban people, the village people did not get the COVID-19 in the initial phase of the disease. So in the first wave, only the city people got the death. A lot of people got the death. But in January 4, 2021, in the second wave, even it spread in the villages also. So this is the vaccine. It is really promising that it is day by day, vaccine population is increasing. So we must do 70 to 90% patient immunized. So this is the symptom. I think you all know this is the Omicron. This is the third wave, which is only we are getting cough and cold, and other symptoms are very minimum. And so this is the journey. This is the last part of my talk. This is the journey I thought for the young scientists, young students who are sitting, still sitting in front of their computer. This is just a journey by the scientists, how mobilization of the global science happened till December 2019, when this mysterious viral pneumonia in Wuhan started. Then by January 20, within a month, they know the virus, novel coronavirus, as a causative for the COVID-19, and they name as SARS-CoV-2. And by 12th January, they already know the genetic sequence of the virus, they know the structure. So by in January itself, they develop a artificial data from the, so that become a first diagnostic testing. And several other companies also started to develop kit in January itself, within a month. Then NIH, National Institute of Health in USA, they actually selected two companies, the Moderna and also Pfizer. These two companies, they actually started to design the viral genetic code, that is mRNA vaccine. They already started in January, soon after they got the genome sequence of this virus. And then Oxford University and German company BioNTech, they also started vaccine designing. And as I told you, that by the April or June, 200 vaccine candidate has been submitted to WHO. So by February, they know the mechanism, that is ACE2 is the receptor for the, which is expressed highly on the human lungs. These are the receptor, and this is act as a key, act as a lock for the key to the spike protein. And by March, they know that started giving the advice to WHO that what are the preventive measures people have to take, like and because they realized that scientists found that from the aerosol coming from the breathing and sneezing and coughing, this is spreading from one human to another. So they advise mask, they advise all kinds of precaution, preventing measure, which is our daily routine nowadays, so I am not elaborating. So then by April, first draft of the over 200 vaccine has been announced by WHO, and by June, there are not, as I said, there is not direct drug, targeted drug against virus, but scientists from their own research laboratory, chemists found that dexamethasone, corticosteroid could be life-saving for the patient. Sometimes back in early 2020, hydroxychloroquine came as a big hype, and our country actually gave help a lot of other country to sending hydroxychloroquine, but later on it was stopped because they found, at least in other country, they found it is ineffective against the virus. So by July, the mode of transmission was expanded, like people, scientists have already discovered that what is the contact surface, droplet, airborne, formite, fecal, oral, bloodborne, even mother to child, animal to human. I told you that that kind of transmission they already deciphered. Then by December 20, Pfizer BioNTech mRNA vaccine, that is the first approval. Of course, the emergency approval came for COVID-19. And December 2020, South Africa find out another new variant, that is the Delta. So then slowly by January, there are a lot of countries were making an agreement to purchase the mRNA vaccine, purchase the vaccine through the COVAX global rollout. But our country, as I told you, our Prime Minister did not sign any agreement to get any mRNA vaccine. So March 21, Delta variant identified in India, and November 21, Omicron variety identified in South Africa. Now it is spreading all over the world. So the virus is really increasing day by day. Don't consider the figure, because this figure is completely changing by each day. So at the end, I must tell that follow still, if you are vaccinated, if you are getting the booster dose, if you are not vaccinated, you have to follow 3 Ws. That is, wear a mask, wash your hand, watch your distance. So this is a very recent paper. This is my last slide. And in the Nature paper, Nature is a very prestigious journal. It is a international journal, and the paper came in 13 January. You can read this paper. In this paper, the summary of this paper, I kept a slide for you, that COVID is here to stay. So country must decide how to adapt. So COVID will be there with us. It is our job to, and then CRISPR-Cas, also a new technology. As I told you earlier, I tried to describe you, and but people are working. Still, scientists are not so successful. They are successful making the diagnosis, but they are not so successful how the system could be used to eliminate the COVID. But I'm sure scientists will one day, he will be successful. Maybe next time I will come to you and tell you the success story. বাট আই উইল রিমেম্বার কবিগুরুজ লেটার, যে দুর্দশার হাত হইতে উদ্ধারের কোনো পথই নেই, এমন কথা মনে করার মানুষের ধর্ম নয়। মানুষের ধর্ম জয় করিবার ধর্ম, হার মানিবার ধর্ম না। সেই জন্য আমরা হারবো না, হারবো না, হারবো না। আমরা কোভিডকে নিয়ে বাঁচবো বা কোভিডের সঙ্গে যুদ্ধ করে জিতবো। থ্যাংক ইউ, বিহেভ রেসপন্সিবলি অ্যান্ড স্টে সেফ। থ্যাংক ইউ ভেরি মাচ for your patience.
Thank you so much, madam. We are really humbled for your valuable talk. Like it was really amazing. So, one question from our side. I'm just showing this on the screen. Madam, the question says, how reliable is the test kit of COVID 19?
Which test kit? RT-PCR. If it is RT-PCR, it is 100% safe. Because as you know, as I was telling, that it has to be amplified, So if it is a very tiny bit of virus is there collected from your nose or mouth, it can be amplified. Maybe it can need to amplify more than 30 or 40. But, that will, CT value will tell you that, even tiny bit of virus is there or not. But that is, RT-PCR is a gold standard. No way you can miss the virus. But other tests like a home kit, like an antigen detection, it is not so accurate. But, sitting at home, you can do it. That is an advantage. So each assay has a advantage and also disadvantage. But RT-PCR is gold standard.
Madam. Thank you. Thank you so much, madam. Thanks for sharing your valuable time with us. Thank you, madam. Now I would request Srimati Sharbari Nath, Associate Professor, Department of English, and also the Secretary, Teachers Council of Women's College, Agartala, to convey the vote of thanks.
Hello, I hope I'm audible.
Yes, ma'am.
Respected resource person Dr. Chitra Mandal, scientist CSIR, IICB Kolkata, President Srimati Manidipa Devarma, Principal Women's College, Organizing Secretary Dr. Subrata Dev, Technical Coordinator Pushpa Ramkhal, Assistant Professor, Department of IT, my colleagues, students, and all listeners present in the webinar. I thank you all for being present in this very interesting webinar. I attribute my heartfelt thanks and gratitude particularly to Professor Mandal, who has taken her valuable time out from a busy schedule and updated us regarding COVID, the hard work behind finding the virus and also its therapeutics. Hope after 80-90% people in India, 80 to 90% people in India are vaccinated, there shall be herd community and we can term these as an endemic rather than a pandemic. I thank all the persons who were behind this webinar and have worked hard to make it successful. Thank you all. I also thank all the scientists all over the world who have been working hard to bring a peaceful and a healthy world to all the people living in this on this planet Earth. I hope that all the scientists become successful, and ma'am, you also be successful and help us to have a
Thank you.
very healthy life. Thank you, ma'am. I hope all to stay healthy and stay safe. Thank you.
Sorry for the convenience. I just want to ask you two more questions, which is from our side.
Sure,
Yeah.
sure.
So the question says, do we require booster dose?
Yeah, actually, this is $1 million questions. Being an immunologist, I can say we don't need booster because when we learned the immunology, we knew these T cells and B cells, all cells has a memory cells. So they should keep their memory intact because you see when we were very little, in the hand we used to take the vaccine, and still in the adult we never go back and took the booster. But you see, we are dealing with the, so we as an immunologist also are puzzled why we need to take the booster dose. But it is true that scientists have observed that something this virus is doing. This virus is very notorious. Scientists has to do more research, more critical research to find out that what exactly they are doing to our immune cells. Because being an immunologist, as I told you before, that we don't should not need a booster dose. But when they are recommending the booster dose, we have to take booster dose because maybe somehow virus is doing something. Therefore, our immune cells become weak or our memory cells become destroyed. Something, but I don't have any hardcore scientific data to tell you that what is happening actually to our immune cells. But this much is true that immune cells are become weakened, and therefore to boost these immune cells we need to do the booster. But as an expert, people who are asking me that why we have to vote, actually why we need to do the booster dose or not, I say no, because I want the people like you, like 16 to that adult who are not getting vaccine, first concentrate on to save our young population. First we have to save our young children, then only we will save ourselves taking a booster dose. So that is my personal opinion. I am not a policy maker, but my personal opinion is put all the money, put all the facility in India, because we have a vast population. We have so many young children who are unvaccinated. So being a mother and being a senior citizen, I appeal that first complete the adult vaccine, the children vaccination, and then you go for the booster. Because we adult already got two vaccines.
Yes,
Am
ma'am.
I clear?
Yes, ma'am. Yes, ma'am.
Okay.
The
What is the
next
other?
question, the next question asks why people are affected by COVID-19 even after two vaccines.
Yeah, as I told you again, it is the actually the question is the same. Like our immune cell is not properly working. Therefore, they are thinking for the booster dose. But if you can see, because we are in very regularly in touch with the hospital and doctor, and they were telling that once you take the vaccine and you are you are taking care of your preventive measure like a mask and everything, your seriousness of the disease, that is not so severe. Like earlier, like if you think about 2020, when it is started in one, that time people didn't know about the preventive measure. So there was I think this little story, if you give me 1 minute, I can share this little story to you, that there is a business relationship with from the one city to Milano city in Italy. So 3 times plane used to go from Milano, that is Italy, to the Wuhan, China. And that time people don't know, cough and respiratory infection and all these things. They didn't know this is due to the COVID. This is due to the SARS-CoV-2. So the travel was continued, and if you remember that death statistics, almost most of the senior people died with the severe lungs disease in Italy. That was the highest death rate in Italy. So therefore, but after vaccination, that kind of serious hospitalization, serious oxygen requirement is less. So that so far that is the data is available with us.
Thank you so much, madam. Thanks for sharing your valuable time with us. Thank you, madam.
Okay.
It
Okay.
was really much inspiring and informative.
I hope some of you become take a scientist as a profession. This is really fun, because once we go to the lab and get a one small positive result, it's like somebody gifted me a diamond. It's more than that. So it's
Yes, yes,
really,
exactly.
really fun. And I would say that scientist profession is a profession যেখানে তোমার স্বপ্নটাকে সত্যি করার জন্য তোমাকে টাকা দিয়ে মাইনে রাখা মানে মাইনে দেওয়া হচ্ছে আর কি। সো ইটস এ ইটস রিয়েলি এ ফান, ইটস রিয়েলি এ নাইস। আই মিন পরের বার আমি যদি জন্মাই আমি সায়েন্টিস্টই হতে চাই।
Ekdom, ekdom, madam. Thank you so much, madam.
থ্যাংক ইউ,
Thanks a
থ্যাংক
lot.
ইউ,
So here the popular talk series comes to an end. Thanks for the patience. Thanks for hearing. Thanks a lot. Stay connected. Have a great day.
থ্যাংক ইউ।